So we started to discuss about the immunity, and in the immunity we discussed that immunity is basically ability of a body to resist the microbial action. Okay, it basically help us from the microorganism. Then we divided the immunity into the two types. The one type we call it the innate immunity, also called as the inborn immunity, also called as the non-specific immunity. And other hand, we classify the immunity into the acquired immunity, also called as the adaptive immunity or specific immunity. We discussed the innate immunity, different types that we seen that innate immunity is due to providing the different barriers, like starting from the physical barrier, physiological barrier, cellular barrier, and that cytokinin. Then we discussed about the acquired immunity and also called as the adaptive immunity. We discussed that in the acquired immunity, the main thing is there will be a memory formation. There will be a memory formation, and the thing is this adaptive immunity, it basically provided by our lymphocytes. Okay, it is provided by the lymphocyte. Fine. Then we discussed that there are two types of lymphocyte called as the B lymphocyte and T lymphocytes. B lymphocyte actually producing what? It is producing the antibody. It is producing the antibody. Whereas T lymphocytes, they are storing, as well as there are four types, and they are also helping in the encounter of the microorganism. Then we discussed about that there are lymphoid organs. Basically the immune system consists of with various immune cells. Apart from that, there is a immune systems. In the immune system, there is something called as the lymphoid organs. We have the two types of lymphoid organ. One is called as the primary lymphoid organ. Another one is called as the secondary lymphoid organ. Primary lymphoid organ is your spleen and bone marrow. Very good. And secondary lymphoid organ is your lymph nodes or lymphatic nodes and spleen. Fine. Then we discussed that. There is something called as the antigen antibody reaction. We discussed. Okay, whenever antigen comes, there will be antibody formation, and that there will generate the antigen antibody complex, which is basically also a basis of ELISA test, enzyme linked immunosorbent assay, which is commonly employed to detect the HIV infection. Now, in our today's class, we are going to first classify the acquired immunity. Okay, and then we will discuss about the immunoglobulin, also called as the antibody. So for classification of acquired immunity, we have the two types of acquired immunity. One is called as the active immunity, active acquired immunity. Another we have is called as the passive immunity. The fundamental difference between the active immunity and passive immunity is that this active immunity is basically developed by a person. When they encounter the pathogens, they develop this kind of immunity, active immunity. Okay, it is basically on effort, a person's on effort. Whenever persons encounter a microorganism, whenever they a person fight with the microorganism, they develop a active immunity. So it is the person's on effort, Now you are seeing that it is further subclassified into the two categories. One is called as the natural active immunity, another one is called as the artificial active immunity. So the natural active immunity, it is purely developed by a person. Yes, it is purely developed by a person. Whenever they encounter a microorganism, they develop a immunity. Suppose as we cited the example of the sebum, that sebum is suffered from a tuberculosis. So when he win the fight with the pathogen, he develops the active, we can say the natural active immunity. Now what I mean by the artificial active immunity? Artificial active immunity means the immunity which is developed by the vaccines. So there is you can say, simple physiology behind the vaccine. The vaccines are basically attenuated or alive or a dead, you can say, pathogen antigen. Vaccines is made up of from the antigen. This antigen is again from the bacterial antigen or a viral antigen. Are you getting my point? Yes? Suppose there is one bacteria, okay? There is one bacteria, and now see, this is the, suppose one human. We can take the example again of the sebum. So he is a sebum, fine. And the thing is, if suppose I am giving him the vaccine, okay? So first we have to understand what is vaccine. So as I said you, that vaccine is either live or attenuated or dead microbial antigen, or bacterial antigen or viral antigen. You know that bacteria is having an antigen. Yes, bacteria have more than one antigen. More than one antigen. Now, these antigens are pathogenic. These antigens are pathogenic, means this antigen can cause a disease. Fine. So whenever bacteria comes in a human body, due to this antigen, there will be a synthesis of antibody. There will be synthesis of antibody. Fine. So there will be antigen antibody complex formation. Antigen antibody complex formation. This much knowledge we have from the previous class. Now I am saying that, what if you can take these antigens, okay, make it less, you can say pathogenic, make it less pathogenic and administer to a person. Yes, if you take the antigen from the bacteria or virus or any pathogens, you take the antigen, you make them less pathogenic, and again you can say administer to a humans, okay, then what will happen? The thing which happens is, when this, you can say, less potential antigen went into the human body, it triggers the immune cells. In turn, immune cell will synthesize the antibody. Now, again in another exposure, if bacteria comes, okay, with the same antigen, our body already have the memory as well as the antibody. So there will be a rapid fire, that is called as a secondary immune response. We discussed primary immune response, secondary immune response. Okay, so next exposure of the bacteria will elicit the secondary immune response, which is more powerful and rapid. They will easily encounter. That is the basis of vaccine formation. Vaccines is nothing. Suppose this is a vaccine. In the vaccine, what is there? Is the antigen. Okay, is the antigen. Antigen can be from the bacteria, can be from the virus. Are you understanding? It can be live in the case of tetanus. In the case of tetanus, vaccines are the live attenuated vaccines. Fine. Attenuation means you are making less pathogenic, okay, less potential or less pathogenic, and you are giving to the any other persons. First time vaccine goes inside human body, it triggers the immune cells, which immune cell is specifically the antibodies. Antibodies synthesize again, there will be a minute reaction. First time, that is called as the primary response. Okay. Now, now this time the new bacteria comes. When bacteria comes, interact with immune cells, our immune cells already have what is antibody synthesized against the particular bacteria. Now there will be a rapid, you can say, reaction between the antigen and antibody, and bacterial antigen is easily destroyed, which is producing basically what? Secondary immune response. This is the basis of vaccines. So come here, we are discussing the types of acquired immunity. We said that there are two types of acquired immunity. One is called active immunity, another one is called as the passive immunity. And the fundamental difference between the active and passive is, active immunity develops by your own effort. It develops by your own efforts. Fine. And the thing is, there are two types. One is called as the natural active immunity, another one is called as the artificial active immunity. The disease you are suffered, okay, and you recovered without any, you can say, treatment or without any vaccines, you develop the antibody, that is called as a natural active immunity. It's a pure person's effort. It's a pure person's own effort. Talking about the artificial active immunity, it is a it is mediated through the vaccines. You can write down the example. Fine. Now, comes here passive immunity. As a name suggests, you acquired from somewhere. You acquired from somewhere. The person which is receiving, they are called as recipient, and the person who is giving, they are called as donor. You have the clear concept, donor and recipient. The thing is, in the natural passive immunity, you are, you can say, provided, come fast, you are provided by your mother with some antibodies, Have you heard about the IgG antibody? Yes, these are the one classes of antibody which you acquired from your mother. Your mother is giving when you are in your, you can say, the fetal life, when you are in your mother's womb. On that very moment, these antibodies, called as the IgG antibodies, it crosses the placenta. Placenta is nothing, it is basically a ultimate, or you can say intimate connection between the mother and fetus, and through this placental layer, or you can say the connection, oxygen, you can say transfusions occur, oxygen and carbon dioxide, fine. Then there will be a food which is going, the blood, basically maternal blood goes inside the fetus. Okay, thereby fetus maintain their physiological requirement. The thing is, along with the food or gaseous exchange, this antibody also crosses the placenta. Which antibody? IgG. So after the birth, till 1 month, you are, you can say, free from the microorganism just because of your mother IgG antibody. Clear? Yes? You need more clarification? We'll have after a few minutes, we'll have the detailed discussion over the antibody. On that time, I will explain you in detail about the IgG antibody, but for a while, you just keep in your mind, the thing is, natural passive immunity means this immunity you acquired from somewhere. Somewhere, basically your mother. Fine? No problem. Now the second thing, the one is called as the IgG antibody. Clear? Fine. The another antibody, after the birth, your mother is like, all of us mother, our mother knows that she have to protect her child. Whatever ways she is putting her effort, and the nature also supported her. And the thing is, this antibody, as I said, it is only functional till the 1 month. But what about the after 1 month? Is there any way to protect? Yeah. When mother is feeding, something called as the breastfeeding. Yes, when mother is feeding her baby, on that time through the milk there will be again one antibody goes to the baby, and that antibody is your IgA antibody. That is your IgA antibody. Yeah. The first milk, which comes from the mother breast, they are called as the colostrum. That is rich in the IgA antibody. Clear? So this is what constitute your natural passive immunity. Talking about the artificial passive immunity, it is something you gain from the artificial antibodies. You are getting something called as the horse antibody, horse pulled antibodies. Okay, horse pulled antibody, or there are other forms of antibody. It is a preformed antibody directly, you can say, a doctor advising you to take, in order to encounter the microorganism, basically the pulled antibodies. Clear? Fine. So, if concluding the acquired immunity, we have the two types of acquired immunity. One is called as the active, one is called as the passive. Active means your own effort, by your own effort, you are developing or synthesizing the antibodies in your body. It can be the two types. Naturally, when you without anyone's effort, your own effort, if you win the fight with the microorganism, your body naturally develop or synthesize the antibody against that pathogen. Talking about the artificial active immunity, if you fail to fight, you are vaccinated, and through the vaccines you develop the antibody. No problem. Talking about the passive immunity, as the name suggests, A donor is someone is there. There is someone donor who is giving you. You are a recipient. Fine. It can be in the two ways. In the natural passive immunity, the donor is your mother, and you are recipient. You are receiving the two, and basically the immunoglobulin are called as the antibody. One is Ig from the during your intrauterine life. I mean the fetal life, crosses through the placenta, and IgA through the colostrum, the milk, first 6 days milk. Fine. Then talking about the artificial passive immunity, it is basically you are getting directly preformed antibodies from the horses or from the sheep. Okay, these are called as the pooled antibody. They are called as the pooled antibody. Write down the one more term, something called as the herd immunity. It was asked in the previous exam, called as the herd immunity. Herd immunity. Okay, herd immunity is nothing. It is just you can say the sum total of all the immunity present in one community. It is a sum total of the immunity which is present in a community. As we discussed, know that innate immunity operate at 3 levels, starting from the species level, then we have the racial level, then individual level. So in a one community, whatever you can say ability to fight with the pathogen is present, they are called as the herd immunity. So if you want to break the chain of infection, or you can say the transmission of infections, you have to strengthen the full community. Full community immunity means basically you are trying to develop the herd immunity in the entire community. You got it? Yes? Fine. Now, so let's move ahead and discuss about antibodies. Till now you have any questions? Yes? You have any question? Response, you have any question? No? We'll move ahead? Okay, fine.